So we spent all kinds of time talking about gas transport. In the last video, we talked about blood flow. Now let's put them together, right? How are we actually transporting oxygen from our lungs into our blood and then getting it into muscle? The way that we do that is by using a molecule called hemoglobin, often abbreviated, and what I'll use most often is HB. So what we do is almost all of the oxygen, 99% of oxygen, is transported bound to hemoglobin. And indeed, oxygen is very difficult to actually be soluble in water or in a liquid, right? So because of that fact, we can't just put oxygen into our blood, right? Since it's not soluble, it needs something to bind to. That binding agent, again, is hemoglobin, right? We actually have two different forms of hemoglobin. One is simply oxyhemoglobin, which is defined as the hemoglobin bound with oxygen. And of course the converse, which is deoxyhemoglobin, which is hemoglobin not bound to oxygen. So as we can imagine, this is going to be found in arteries, and this is going to be found in veins, right? So if we can do the mass, so the amount of oxygen that transported per unit of volume of blood is dependent on the hemoglobin concentration. So each molecule of hemoglobin has what we call 4 sites of heme. These are iron type, essentially have an iron backbone in this hemoglobin that is able to reversibly bind oxygen loosely, right? That reversibility is key because we need oxygen, the ability to come in and bind and also be released in tissues, right? Each heme molecule can transport 1, and red blood cells or hemoglobin have 4 of these heme sites, right? And we can say that each gram of hemoglobin can transport about 1.34 mils of oxygen. And then if we estimate that males have about 150 grams of hemoglobin per liter of blood, multiply that by the amount of oxygen that each gram can carry, that each male can carry about 200 mils of oxygen per liter of blood. And so you say, okay, why is this important? So one of the reasons, as we relate it back to exercise physiology, is the idea of blood doping. The goal of blood doping is actually done in one of 2 ways. One, they'll take a drug called EPO or EPO, erythropoietin, which stimulates hemoglobin production in the body to produce more red blood cells. So therefore, this 150 grams of hematocrit or hemoglobin gets higher, you then can increase the amount of oxygen carrying capacity. The other way, which became even more popular since they started testing for these foreign substances, is to actually take blood, spin it down, keep the hematocrit, which is made up of red blood cells, re-inject the hematocrit in, and use that to boost red blood cells. Again, the idea is to give more carrying capacity to these heme groups located on hemoglobin. The more carrying capacity, the more hemoglobin, the more oxygen we can pick up and deliver into the muscle. So let's take a look. So again, here's our hemoglobin molecule. We have 1, 2, 3, and then kind of hidden in here is a 4th site with these heme or iron-based units that are important to reversibly bind oxygen. Hemoglobin is a really interesting molecule because it doesn't just act as, oh, pick up and give off whenever I feel like, right? It has that idea of reversibility is incredibly key because different environmental factors or things in the body are actually going to affect the dissociation of oxygen to the heme group in oxyhemoglobin, right? So what it is essentially going to detail is how much oxygen are we going to be unloading and dumping off at the tissues, and it's also going to determine if we're going to pick up or drop off, right? So if we look at what the PO2 in arteries, right? We covered this earlier, but this is about 95 in the arteries, right? get a, so PO2 equal to 95 in the arteries. So as you can see, when the PO2 is very high, the percent of saturation of hemoglobin is nearly 100%, right? So what that means is at a high PO2, or in our lungs, for example, which is 104, that the hemoglobin becomes fully saturated. All four sites are going to be carrying oxygen. Hemoglobin is now going to be converted into oxyhemoglobin by the binding of oxygen. So this is important. It's also important to note that maybe if we have some type of disorder, some type of VQ mismatch, as we talked about in the last lab, is, if we're not getting adequate ventilation, maybe our PO2s are low or we're getting low blood flow, what are we going to pick up? So you'll notice that this curve is actually very flat at the top, right? We have a near plateau. So this is interesting that even if we have low ventilation, that we still get very high oxyhemoglobin saturation. In fact, clinically, anything below 80 is considered very severe and requires medical attention. And so you can see that to get below 80, the PO2 in the lungs would have to be around 50. Right? At that point, if we were getting that low of ventilation, then our VQ mismatch would be that low, then we would no longer be able to adequately saturate hemoglobin, right? So that's really important, right? It doesn't take a very high PO2 to saturate. But then we have a sharp decline, or depending on the way you look at it, this figure says a sharp increase, because this is talking about it at the tissue level, right? So the tissue level at normal, at rest is, as you'll recall, is 40 millimeters of mercury, right? So we can look at this value and say that that at a PO2 of 40, we actually drop off about 25% of the oxygen in our blood into the tissue, specifically as we talk about exercise and metabolism, talking about muscle usually, right? So again, once the PO2 drops, then that affinity for hemoglobin and oxygen is going to decrease, right? When it decreases, then that reversibility comes into play and it will give off oxygen. So again, to review, at the lung, very high PO2 equals formation of oxyhemoglobin, or loading, again, 95 to 104. So we load at almost 100%, and then at tissues, low PO2s of 40, that causes us to then release oxygen to the tissues. So the next important thing that we'll talk about is what happens in exercise, right? So here's normal. Again, we'll dump off about 25% of our oxygen into our tissues at rest. The PO2 at rest in tissues is about 40. So again, as we move from arterial to venous, the PO2 is going to decrease. We're going to dump oxygen, converting from oxyhemoglobin to deoxyhemoglobin. That is, again, we can say it dissociates. Right? If we look at the difference between PO2, arterial, and venous PO2, we'll see that exercise has a very large effect. Right? We've talked about this in the cardiovascular. Arterial PO2 is not going to change for most people. Right? So we're able to keep that PO2 at the lungs. This is, again, kind of an average of the lungs and the arteries. Then our arterial content isn't going to change because our arterial PO2 is the same. So we're going to dump and fully saturate all of our hemoglobin here. However, our venous PO2 is going to be much different, right? So in this case, our muscles during exercise are consuming lots of oxygen. We're taking lots of oxygen. Therefore, the partial pressure that oxygen is exhibiting is much lower because there's just much less oxygen. Less oxygen, less PO2, right? And so if we look at the venous oxygen content, we can see that we take a lot of oxygen out of the blood during heavy exercise. And we discussed that. Remember, AVO2 is increased incrementally with exercise training, so that we can say at rest about 5 mils per deciliter of blood, whereas when we talk about heavy exercise, the AVO2 is about 15 mils per deciliter or 100 mils of blood, which is how we calculate AVO2 difference. So what that does is it creates a stark contrast in the venous PO2. So here in green, again, we've shown that this PO2, when it decreases, here we have it marked at about 18, right? At 18, because of this very sharp curve, we'll see that this sharp curve then means that we dump off, look, 75 to 80% of the oxygen due to this low PO2. Remember, at rest, we only dump off 25% of the oxygen leaves the bound hemoglobin and goes into the tissue. At heavy exercise in the muscle, because of this stark decrease in PO2 due to oxygen consumption, we now have a PO2 of about 18 to 20, which means the sharp curve says that we're going to dump almost all, about 75% is unloaded during heavy exercise to provide more oxygen to working tissues. So that's one important feature. This nice curve dictates that as PO2 differences increase, as we increase that difference in change, oxyhemoglobin is going to dissociate, dump more oxygen, and become deoxyhemoglobin. So that's one way. So as we have discussed, one of the byproducts of metabolism is altering the pH in muscle. This is another way that we can affect this oxyhemoglobin dissociation curve. Right? We'll just say this: with a decrease in pH or high acidity, we decrease the affinity of oxygen to hemoglobin. Hemoglobin wants to give off more oxygen. If we look at this curve, our normal pH in muscle is 7.4. Okay? As we continue to work out, we produce hydrogen ions. There's also some other acids that we're going to discuss in the future that drive pH down. We become more acidic, which takes our pH from 7.4 to 7.2. Right? So what does this do to the percent of oxygen bound to hemoglobin? Right? So if we take numbers, as we talked about earlier, we'll take numbers from the previous slide. We did 18% earlier, but we'll do 20% here. So if we draw a line up from 20%, I am not the world's best artist here, especially not with a mouse. Right? So again, during heavy exercise, our AVO2 difference goes up. We're consuming lots of oxygen. Partial pressure goes way down from resting values of 40 to exercising values of 18 to 20. So here we're going to use 20. So if we look at this curve, at a PO2 of 20, we'll see that there is the dissociation curve states that only about 33% of oxygen bound to hemoglobin will remain. The rest will dissociate, right? So in this situation, normal rest, a PO2 of 20, we can say that 66% of O2 goes to muscle. Okay? But let's look at, because hemoglobin is an enzyme, just because it's an enzyme and a protein, it is affected by both acidity and, as we'll see next, temperature. When acidity affects it, it actually decreases the affinity of oxygen, which is beneficial, right? So here we can draw our line here, and we can say that at heavy exercise, the affinity for oxygen bound goes down. Therefore, we release more into the blood or into the muscle tissue for oxygen consumption. So we'll now see here that only about 24% of oxygen remains bound to hemoglobin. The rest is given off. So in this situation, 76% of oxygen, due to the pH's effect on hemoglobin, is given off, dissociated into, and able to be transported into the muscle to be used for oxidative metabolism. We call this the Bohr effect, and it is a very strong influential way that we can get more oxygen to our muscles in times of need. Temperature also, or before we move on, I'll just say this. The opposite change in pH will have the exact opposite effect, right? So if we have a leftward shift, if we become more basic, which doesn't really happen at least in muscle physiology, then oxygen actually becomes tighter, more tightly bound. Therefore, for any given partial pressure of oxygen, less oxygen will dissociate. The exact opposite of what we see with exercise and muscle. If we look at what happens with temperature, in general, the more we work out, again, our energy systems are an inefficient process, so we don't use all the ATP that we generate for muscle. For movement, we also create heat, right? So our muscle tissue, average body temperature is about 37 degrees Celsius. As we exercise, it can approach anywhere between 40, or in this extreme example, 42 degrees Celsius in the muscle temperature, right? So blood temperature causes, again, a rightward shift. So if we increase temperature, we see a right shift in this, right? So again, similar to pH, a right shift says that for any given PO2, we will actually release more oxygen into the blood. More oxygen will be dissociated, and the percent saturation will be less. And of course, the opposite happens, right? At lower temperatures, it will give off less oxygen. More oxygen will remain bound to hemoglobin at that time. And you may say, well, does this affect anything in the lungs if we're changing pH or temperature or anything like that? Remember that this curve plateaus very high up here. So even a rightward shift, we're still going to have very, very good transport and saturation level at the lungs, even if we have any type of these effects. Again, just because of the S shape of this curve. Plateau up here says we got no problems. We can saturate fully in the lungs really easily. These are more affected by these steep slopes during exercise. So the last part that we'll look at is myoglobin. Myoglobin is the oxygen-bound protein in skeletal and cardiac muscle. So this is actually how we transport oxygen that gets from the hemoglobin into skeletal muscle, and then from the muscle into the mitochondria, so that it can act as the final electron acceptor in the electron transport chain. Right? It is also the reason that it gives off a bright red color. Right? Myoglobin, when bound to oxygen, becomes higher color in red. It has a much higher affinity for oxygen than hemoglobin, even at a low partial pressure of oxygen. So what we see here is a leftward shift in myoglobin compared to hemoglobin, right? It means it wants to bind oxygen much more. And this makes exactly sense from a physiological perspective, or at least I hope it does, in that you want hemoglobin to dump oxygen, and you want myoglobin to grab oxygen from hemoglobin, right? So it has to have a higher affinity for oxygen than hemoglobin in order to grab it away, right? We're only going to move from higher partial pressures to lower partial pressures, and so myoglobin is going to grab it, right? This allows myoglobin to store oxygen and even reserve it in periods of time from rest, right? We talked about low muscle oxygen levels when we discussed EPOC. This is the idea, is that myoglobin binds oxygen and we can replenish those stores. So again, must be recovered, as we talked about in class with EPOC during that oxygen deficit. If we look at fiber type specific, type 1 muscle fibers, of course, more oxidative, are going to have more hemoglobin than type 2A and type 2X fibers, which again is what gives them those redder colors. I think the video earlier on talked about it, and I didn't mention this in hemoglobin, but that's one of the important things, or one of the things to note about hemoglobin. The more oxygen bound to hemoglobin, it turns bright red, which means our arterial blood gives a bright red color. As it gives off oxygen, it actually looks more of a bluer color, right? And so that's why our veins are considered blue, right? So in this situation, we can put more oxygen that can be stored in muscle in the myoglobin, and therefore type 1 are much more red. So we spent a lot of time talking about oxygen, but the other important factor of the pulmonary system is the ability to transport carbon dioxide out of tissues and then into the lungs, where it is expired and gotten rid of as waste. there are 3 different mechanisms of CO2 transport in the blood. Unlike oxygen, which has almost no solubility in blood, CO2 does have a small level, so we can dissolve some CO2 in plasma, and about 10% of CO2 is transported that way. Second is CO2 can actually combine with hemoglobin. It doesn't bind nearly as strong, doesn't have near the affinity of oxygen, but remember, in low PO2 levels and higher PCO2s, remember PCO2s in the tissue, about 46, PO2s can be 40 or less, then CO2 will combine to form a form of hemoglobin called, I don't like saying this one struggles, carbaminohemoglobin, right? And that's about 20%. However, that's not much CO2. The majority of CO2 is actually released and carried as a carbonic acid or in the form of bicarbonate, right? So let's spend a little bit more time on that. This reaction takes place in the red blood cells. So carbon dioxide goes from the tissue level. Remember a PO2 here of 46, or PCO2 of 46, and a PO2, PCO2 of 40. Right? So CO2 will go out, down its pressure gradient, and then into red blood cells here. So we're following this tracing here. Right? The red blood cells have an enzyme called carbonic anhydrase, which says at high PCO2 levels, it causes carbon dioxide to combine with water to make H2CO3, otherwise known as carbonic acid. Inside this red blood cell, carbonic acid actually then splits or dissociates into a hydrogen ion and HCO3, which is bicarbonate. This hydrogen ion will ultimately bind to hemoglobin. Bicarb will actually go out of the red blood cell. Since bicarb is negative, if it were to go out without having anything come back, then we would have some type of electrochemical imbalance. So we actually have a chloride ion known as the chloride shift replace this removal of bicarbonate. So we now have our hydrogen bound there, and then we'll move to what happens in the lung. So when blood reaches the lungs, the PCO2, sorry, there's a typo, the PCO2 is greater in the red blood cells. So we have several different factors. One, again, our normally stored CO2 just goes out. We have about 10% in our plasma, about 20% here bound as carbaminohemoglobin. But we also have this this formation of this extra hydrogen ion, right? So what happens when we look at this of what's going on is that, again, oxygen is going from high PO2 to low PO2, right? So very high here, 104 for our PO2. So that's going to come in. It's going to have a higher affinity, as we just talked about, and it's going to bind to those heme groups, which causes the hydrogen ion to be released from the hemoglobin, and then hydrogen is able to, again, with the help of enzyme, convert back to the bicarbonate form of carbonic acid, which can then be converted into CO2 and water. CO2 is then expired, or sorry, skipped a step. CO2 then is greater in red blood cells, so finally CO2, which comes out, is expired and diffuses out of the red blood cell into the alveoli and then leaves with expiration, and that's how we blow off CO2 from metabolism that we are creating. So in summary, about 99% of oxygen is chemically bound to hemoglobin. Again, very little saturation ability of oxygen in the blood. It needs some type of bonding agent, and hemoglobin is that. So the effect is of partial pressure on the combination of oxygen with hemoglobin is illustrated by the S-shaped curve. remember? So it doesn't take a really high PO2 to fully saturate oxygen, and then it's very steep in the partial pressure seen at tissue. So at 40, we give off about 25%, but in times of high oxygen needs, where PO2 decreases even more, it dumps large percentages, 75 to 80% of its oxygen there. An increase in temperature or a change in pH shifts the curve to the right. That shifting of the curve means that for any given partial pressure of oxygen, more O2 will be dissociated from hemoglobin and given off to be into the tissue. And then last but not least, we talked about carbon dioxide and its three transport mechanisms. 1, directly dissolved in plasma. 2, bound to hemoglobin in carbaminohemoglobin. But 70% is essentially formed through bicarbonate, right? Where bicarbonate is formed, we then transport a hydrogen molecule that we then was in that bicarbonate all the way to the lungs. We then release that. When oxygen binds, that hydrogen becomes bicarbonate again. Reverse reaction then takes place of converting it back to CO2 and water and blowing off CO2 in that effect. So that ends this module, and then tomorrow we'll talk about this in response to exercise.